Sanger sequencing is a method of DNA sequencing based on the selective incorporation of chain-terminating deoxynucleotides in the process of DNA amplification. A set of gold standard reference genomes for Arabidopsis, rice, sorghum, and maize were first sequenced using reiterative Sanger-based approaches along the minimal filling path of bacterial artificial chromosome or BACs. Genome sequence of a plant provides a mean for understanding the genetic basis of the differences between plants and other eukaryotes, and provides the foundation for detailed functional characterization of plant genes.
Now we will mention the process. The first step, you need to amplify the DNA segment. The next step is to add heat to the sample to denaturalize the DNA at 82 degrees. So the template strand will be the 3' to 5' and a primary is added. In order to identify the chain-terminating nucleotide, we add a specific fluorescent dye into 4 vessels. When you have your primer DNA, you add a DNA polymerase. You add a DNTP that lacks a hydroxyl group in the third carbon. So in here, the DNA polymerase will attach the DNTPs. But in order to stop the DNA polymerase, the DNTPs will achieve this goal.
DNA fragments of different lengths are formed in the vessels. Polyacrylamide gel electrophoresis is used for the DNA sequencing. Each vessel is verted on one specific site of the polyacrylamide gel. Then, the sequencing is read from the bottom of the polyacrylamide gel. This results in a complementary DNA sequencing of the DNA sample.
Now we'll talk about the applications in agronomy.
Resequencing application. Sequencing of DNA followed by comparison to known or reference sequence. SNP phenotyping. Detection of 1 to 10 single nucleotide polymorphisms. Microsatellite ISSR analysis. Intersimple sequence repeat. Analyzing is a technique commonly used for identification of individuals, mostly plant species, based on variation found in the regions between microsatellites.